And, oops, wrong button. Screen share. Actually, I think that we stopped maybe one slide earlier. Okay. Does this look about right for y'all? Trying to pull up my thing here where I can see everybody. Okay. So photosynthesis wise, do y'all have any questions for me? Light dependent reaction. Y'all want to wait till we get the quizzes back before we go over that? Okay. Was that a surprise on the quiz as a bonus question? You have any thoughts? I'd love to hear from you. Anything?
I kind of knew it was going to be on there, but I stopped at like the NADPH.
Okay. Did you mention the ATP aspect?
Yes.
Okay.
Yeah, the centrage.
And those are the main products of the light dependent reaction, so that's good. We're gonna kind of hit the light independent here in a second. I think I've got it coming up. Yeah. So this diagram in the next slide is very similar to one we've already seen. You know, we've got water being broken apart into oxygen. We make some ATP. We give off our electrons, and we create some protons, H pluses, that will go into some of this internal membranes. Our photosystem is hit by light. It gets excited. And just for a second, I wonder if I can switch to screen. I want to go straight to the whiteboard here. So can everybody see the whiteboard? Okay, so here's your photosystem. So PS, when light hits this, the way I just kind of view this is that the electron goes up in energy, and then there's a kind of a stair-step process in which we lose energy. So there's a number of steps along the way: plastoquinone, cytochrome B, forgetting another one off the top of my head, but as they stop, maybe I shouldn't say stop, as they get transferred from one thing to another along this pathway, what do you think is happening to the energy based on my diagram?
It's getting lower.
Yeah, it's getting lower. Okay, and so how do we finish this process to get over to NADPH? Well, about the time that it gets too low to do much anything else, it ends up reaching the next photosystem. What do you think happens there? Kyra, I'm going to pick on you. It repeats. What do you mean by it repeats? By the way, perfect timing. I called on you right as you put that in. Okay, starts the whole process of when light hits it and then the energy goes up. Exactly. So what goes up? It's the energy level that kicks out the electron, and it stair steps a little bit more. What do we end with? That electron ends up in the NAD. Always leave out the D. There we go, NADPH. I say it, but I don't write it. I don't know why. So even when I type it, I leave the D out. Okay, so we might end up with this electron here. So I like drawing this stair-step process because it helps me to understand that energy goes up and then it slowly works its way down and then it has to be increased again by more light. And I guess I should draw that in. There we go. How do y'all feel about this process? Y'all okay with it? Okay, so let's switch back. So, oh, plasticine, and that's the third one I was thinking of. So electrons go up, it gets transferred to plastoquinone, cytochrome, plasticine, and then we're back to the photosystem. And then it goes up, and then it's ferredoxin, flavor protein, and then it ends up in the NADPH. Okay. I don't need y'all to go into great detail on this, but I need you to understand the kind of general idea of how photosynthesis works. That's what I'm going for. So this is the light dependent part of the reaction because you've got to have light. Now the light independent in more depth is the Calvin cycle. And I like this name a whole lot better. Sometimes you'll see it called the Calvin-Benson cycle than saying light independent because it truly is not light independent. If you want to learn more about this, take plant physiology with me and we will definitely cover that. You can pick on Elena. I'll pick on her because she's had plant phys with me already. We go into photosynthesis for like 3 weeks, I think it is.
Yeah, about 4.
It was a long time, yeah. So basic ideas here. Carbon dioxide comes in. Where does carbon dioxide come from for plants? Where do they get it from? I'll pick on Haley. Is that okay?
The atmosphere.
Atmosphere. Awesome. Okay, so plants absorb carbon dioxide. In fact, there's 6 of them. That's where they get these 6 carbons. And they'll combine with 6 molecules of RUBP with the aid of an enzyme called rubisco. And rubisco is extremely important. So might you need to remember rubisco for the rest of your scientific life? I would say yes, partly because rubisco has been claimed to be the most prevalent enzyme on the planet. Okay, that's just how important it is for how life works on our Earth. So these things come together, 6 carbon dioxide, 6 RUBPs, they result in a 12 and 12 PGA molecules. And so we've got these 3 PGAs, there's 12 of them. And you don't have to worry about what a PGA is, it's just an intermediary molecule. We take the energy that's been stored in NADPH and ATP from the first step of photosynthesis, and it supplies the energy and electrons to reduce 3-PGA to 12-GA3P. Okay, so we're creating these new molecules. Now, when I say reduce, how on earth does it make any sense from 3 to 12? How is 3 to 12 a reduction? Can anybody give me a clue? Is anybody confused by that? Like, you can just raise your hand. Okay, does anybody remember chemistry? So remember the redox reactions we talked about? So did we talk about Leo says Ger? Okay, so let's go for this. Leo, what does Leo stand for? L stands for loss. Okay, so we are losing what? Loss of What's the
Electrons.
loss of electrons? Okay, is sorry, loss of electrons, Leo, is oxidation. So what is the GER? G
Gain of electrons.
is, what's the R stand for?
Reduction.
So now do we see how reducing goes from 3 to 12? Weirdly enough, reduction is an addition of electrons. Now, does anybody have their mind just kind of go, whoa, what? How was adding electrons a reduction? What is the charge of an electron? Is it positive or negative? Y'all know that's right.
Negative.
It's negative. So, okay, so if we're adding more electrons, are we reducing the charge? Does that help to make sense? That's how I keep it straight. So when you see a reduction here and you're going, how do you go from 3 to 12 and that's a reduction? It's because we're adding more electrons, okay? The charge is, increasingly negative. Okay, so that's what I think of, at least, how I keep that straight. Now, we make 12 if everything goes well. 10 of those 12 get turned back into RUBPs. 2 of them are extra and that they get kicked out of the cycle. Let's see if I can get to the cycle. So they get kicked out of the cycle, not as a bad thing, but actually as food. So let's start here. Where's the CO2? Okay, here's the CO2 at the bottom. The cycle begins here. We've got our RUBPs. We bring those together to make 12 3-PGAs. Okay, and if you want to, we could even go through and count the carbons of all these things. No, we're not going to, but we're just saying we could. And that would explain a lot of the numbers that we're seeing. We're actually seeing, if you've got 6 CO2s, how many carbons do you have? You have 6. We'd have to go in and look at the molecular formula of the RUBP to see the carbons there. We could count carbons, but that's more than I really want us to get into. I'm just saying that we can. But when you combine these two, you get your 12 3-PGAs with ATP dropping off electrons. We go around this cycle with NADPH dropping off electrons. We go around this cycle and we end up making 12 GA3Ps. These GA3Ps, also sometimes called PGAL, and like, I don't know, Dr. Welch may call them PGAL in her class, like when you take ecology. I mentioned, I think, it's a PGAL in my Bio 2 class, but they have both names. We say we make 12 of those. 2 of them get kicked out if we make all 12 of them. If you could take plant phys, you'll actually see that we don't always do that. But under the ideal situation, we make the 12. 10 of them get returned and recycled back into RUBPs. 2 of them get kicked out. These 2 extra are basically the reason for doing the Calvin-Benson cycle. If you don't make this, then you're not making food. Okay, these go on to become glucose, starch, and anything else. Does that make sense for everybody? Okay, so the whole point of this process is to make these 2 molecules so that we can make food that the plant can survive on. Okay, so we went through the first step of photosynthesis and the second step. Do y'all have any questions? I'll take that as a no. Okay, so what happens if photosynthesis can't go as planned? So for example, if it's really hot outside and you can't open your stomata, which are these little features on the outside of the cell, outside the leaves, little cellular, minor, very, very small, minuscule cellular structures. If it's really hot, the stomata will stay closed. and you won't absorb carbon dioxide. Well, if you can't do that, can you do the Calvin-Benson cycle? No, because you've got to have carbon. So what they do is they'll sometimes switch over to using oxygen instead of carbon dioxide. In fact, oxygen is fixed by Rubisco more readily than carbon dioxide is. It has a bit higher affinity for oxygen. And the reason normally that we don't have Rubisco using oxygen is because we have flooded it with carbon dioxide. So carbon dioxide becomes more prevalent, but that's not the case if we close up the stomata. So think about this for a second. If you close the stomata, will there be a lot of oxygen left over? Think about the first step of photosynthesis. What's the main waste product of the first step of photosynthesis? You break that water, and what's one of the things given off that the plant doesn't use?
Oxygen.
Oxygen. So if you close the stomata, you have a buildup of oxygen inside the plant, right? So we have this process where oxygen becomes more available. Rubisco really likes to attach to oxygen. And so plants kind of survive very hot, dry conditions where they don't open their stomata. They can retain their water for a while. And so they end up basically going through photorespiration. So they use light, but then they do something more like respiration. And it involves the chloroplast and the mitochondria and an intermediary between the two called the peroxisome. So we won't go into great detail of that, but the idea is that plants can handle this, but it's not ideal. This is very expensive, and it's, for what it's worth, it's not enough energy for maintaining the plant indefinitely. Okay. There are some other specialized things that we need to keep in mind because it will affect some of the plants we talk about later. So what we have described up to this point is the first step of photosynthesis and the second step of photosynthesis. And for all intents and purposes here, that is all of photosynthesis for something called C3 plants. So the photosynthesis that we have described up to this point is C3 photosynthesis. Now I'm going to real quick go back and show you something. Can you imagine why it's C3? 3 PGAs right there. C3, it's 3 carbon molecules in photosynthesis. Now, there is a way to make something with 4 carbons, and we have specialized structures in these plants. So things like grasses, the grass family, have this C4 photosynthesis or a 4 carbon pathway. So I usually will refer to this as C4 photosynthesis. Okay, C4 plants have Cran's anatomy. They basically have large chloroplasts with very, very few grana, if any. And they all are around the bundles, around the veins. They have some smaller chloroplasts, very well-developed grana throughout the rest of the leaf and the various mesophyll cells. The thing is, 4 carbon photosynthesis is more expensive. It requires 5 ATP instead of 3 ATP. So C3 photosynthesis is definitely the way to go if you're just looking at it from an energy perspective. Okay. The reason why we have C4 plants is we have situations where there are really hot and sometimes dry situations for plants. Think about maize. Maize and corn. Maize is the same thing as corn. Oh, just a little side note on an English thing. It's kind of cool. Does anybody know why we have different names like maize and corn? Does anybody know where the word corn comes from? In older English, corn used to have the concept of any kind of grain. And at some point, when Europeans encountered maize, the word corn, of course, was applied to it because it's a grain. And I don't know the reason exactly, but corn seems to have gotten stuck with specifically maize at this point. So the scientific name for corn, what we think of as corn, is called Zea mays. So maize is its probably more specific name. But maize is a grass family plant. Same thing with sugarcane, sorghum. They grow in the tropics that are usually really high temperatures. And so they have to deal with these high temperatures and it affecting their water loss. Okay. So a lot of times they have to deal with, oh, great, I'm going to lose my water if I'm not careful. I can't do photosynthesis like the rest of these plants in cooler areas. So what they do is, here's the bundle, the vascular bundle, to be more specific. Okay, where the water comes in and the photosynthetic product goes out. And then we've got these large cells around the bundle. They're called bundle sheath cells. And those bundle sheath cells have the Cran's anatomy. They have very few grana. Their purpose is not to absorb light so much. Their purpose is to do the second half of photosynthesis. Okay, the light independent, or better yet, Calvin-Benson cycle. So what happens is that carbon comes in through the stomata. That's what these little things down here are, opens, and they can open and close. Carbon can come in, and then off to the right here, you can see that carbon gets turned into something called malic acid. Malic acid then gets sent to these bundle sheath cells and concentrating the carbon dioxide in these bundle sheath cells. So what we have is a situation where we try to keep the oxygen and the carbon dioxide from interfering with one another. Remember, in C3 problems, when you have photorespiration, it's because you close your stomata and you end up with too much oxygen. Well, here, when you can't get as much carbon because you're not opening and closing your stomata as much, what do? Well, you take that carbon dioxide and you concentrate it so that you get large amounts of it, but in small areas. And in this case, the small area is the bundle sheath cell. So you have more carbon dioxide than oxygen there. And so you do the second half of photosynthesis in the bundle sheath cells, and you do the first half of photosynthesis in the mesophyll cells that are just out and about. So what happens with C4 plants is that they divide their photosynthesis into two locations. Whereas C3 photosynthesis does both parts in the same cell. Here we've got it done at two separate cells. Mesophyll for gaining the CO2, and then we put it into the malic acid, and then the malic acid drops it off over at the bundle sheath cells so that they can do the second half of photosynthesis, the Calvin cycle. Is anybody lost? So we got one more kind of photosynthesis coming. Do you think you could describe the difference between C3 and C4? Okay. So here's two questions that I would leave you with to try to kind of like test yourself. One, describe the difference between C3 and C4 photosynthesis. And two, can you describe how they function differently? Okay, so what is the difference and how do those things function differently? So I've already kind of mentioned this, but I'll repeat it. Those plants that do C4 with the Cran's anatomy, they produce the oxaloacetic acid. And I'm not worried about you remembering oxaloacetic acid, okay? That just happens to be the 4-carbon compound for C4. I'm not even worried that you remember the PEP or how the carbon dioxide is combined. What I want you to remember is that it gets absorbed and combined in the mesophyll cells, and then it moves them through malic acid to the bundle sheath cells for the purpose of reducing photorespiration.
Can you repeat that? It gets absorbed in what in the mesophyll cells?
So, let's see if I can repeat that exactly. So, carbon gets absorbed in the mesophyll cells into a 4-carbon compound and then transferred through the form of malic acid or malate. You may see it sometimes called malate, to the bundle sheath cells.
Okay.
So that you can then do the Calvin-Benson cycle. And so that we end up with, the ability for the rubisco to absorb carbon dioxide because there's more carbon dioxide available. Why does this matter? Well, depending on where you live on the planet, if you're a plant, there are places on the planet where it is more energetically useful to be a C3 plant versus a C4. We live in an area where, depending on the time of year, it's better to be a C3 plant versus a C4. Have y'all ever noticed that your yard composition seems to change throughout the year? Early in spring, you see all these nice broadleaf kind of looking plants, different kinds of weeds, a lot of times with different flowers. And then as it gets hotter in the summer, you basically only see grass. Have y'all noticed that? Anybody actually? I think Akiri is kind of saying, yeah. Do y'all pay
Yes.
okay. Do y'all pay attention to your yards? Okay, if you mow, you probably do notice that kind of stuff, especially, or so what happens is for us through the year, it's better to be C3 at the beginning of the year when it's cooler, a lot more water, a lot of rain. And then as it gets hotter, we see more of the C4 plants kind of sticking it out. Early in the year, C3 plants have the advantage. And so they kind of overrun the C4 plants. And then it switches. And that's what we're seeing with this graph here on the left. On the right, what we notice is that as you increase temperature, we have the same thing. So on the left, it was light intensity. On the right is temperature, which usually are correlated with each other. As temperature goes up, C3 plants really don't do well. On the other hand, C4 plants don't really change. Like, it doesn't matter if the temperature is kind of low or high, they are equally okay. So when it's a low temperature, C3 plants have the advantage. When it's a high temperature, C3 plants lose the advantage because C4 plants are just kind of like, ho hum, we're okay. Do you see any applications for climate change here?
Yes.
So what do you notice? What do you think?
As it gets hotter, we're probably gonna see more grasses instead of C3 plants.
Yeah. One small problem with that is most of our crops, not all, if you like apples, if you like strawberries, they're going to have a hard time in a hotter world. Okay. Sugar cane, probably okay, but not sugar beets. You see what I'm getting at? I mean, it's going to have an economic effect on us if we're not careful. And trust me, we're not being careful. Okay. You'll see summertime lawns having certain kinds of like crabgrass, and especially up a little further north, Kentucky bluegrass is when we mentioned it. And I put this in there because I took botany in Kentucky. But a lot of these grasses are similar across the eastern US. What's really cool is that C4 as a method of doing photosynthesis seems to have developed evolutionarily more than once. So it is a situation where you've gotten convergent evolution a number of times. And it's only found in angiosperms. Angiosperms are your flowering plants. We do not find C4 so far in gymnosperms, which are your pines and conifers. We don't see it in the ferns, the mosses, or any of those other ones. Okay, we don't see that. Okay, let's talk about CAM. CAM is my favorite. I just like this one so much. So CAM stands for crassulacean acid metabolism. You don't have to remember that. And crassulacean deals with the family of Crassulaceae, where I think it was first discovered. It's not the only family that has CAM though. CAM is also a C4 photosynthesis, okay? But it does things differently, so we call it by a different name, call it by CAM. What it does is something very similar to C4 photosynthesis, but instead of breaking things into two different locations, mesophyll cells and bundle sheath cells, it does things at different times. It does things at night and then during the day. So it accumulates its carbon through the form of carbon dioxide at night when it's cooler. When it's opening its stomata means it won't lose as much of its water. And then it does the rest of photosynthesis, actually all the photosynthesis during the day. It just gets all of its carbon at night, stores it until the day. So they can really, really, really do very, very, very well under very limited water. So can you think of some plants that do well without being watered a lot? We have a certain term that we use for some of these plants.
Succulents.
Succulents. Yeah, a lot of succulents are CAM photosynthetic. So what they do is you've got your chloroplast. Your absorption of carbon dioxide is done at night, and then everything else is done during the day. The light and the Calvin cycle steps are there during the day. But they don't open their stomata to get the carbon dioxide. They've done that already. And they store it as malic acid. Just like the malic acid was a transfer from one cell to the other in C4, malic acid is a storage molecule. In this case, it's transport and storage. It gets stored as malic acid overnight in the vacuole of the plant cell. And then it gets removed into the regular cell. So no real big major details other than night and day. Their chloroplasts do not have a different anatomy like the C4 plants do. They look just so like C3 plants. And then this to me is the most fascinating aspect of all. CAM plants can switch to C3. So they can be C3 when they need and CAM when they need. I find that just absolutely fascinating. Think about it. If you don't have to use the extra energy to do that, why would you? Makes no sense, right? I mean, if you have, I mean, do you just go spend more money on some clothes than you had to? No, you're going to do it on sale, right? Yeah, and then that's what they do here. They only spend the extra when there really is no choice. So they can really function well under low water conditions, or they can actually function well under high water conditions too. And I'm pretty sure that a lot of things in the greenhouse only survive because they're probably cam photosynthetic, because there is some major problems in there from time to time. Okay. Now, you might see this pineapple here. This pineapple is as a cam photosynthetic plant. I think there are some cacti. There's some really cool stuff out there. Excuse me. Stone crops, which is the plant that you're seeing there. You may have seen them. People grow them really commonly in pots and sometimes on the ground. These are sedums. Orchids can do this, not necessarily all of them, but many of them. So CAM is really fascinating. Me personally, I think CAM might be the way that we survive, well, maybe plants survive global warming. I'm not really sure if humans are going to make it. Yeah, I don't know. Okay, we're briefly going to mention respiration. It's not plant specific, so we're just going to bring it up, but not go into great detail. You cover that in a number of other classes if you haven't already. Respiration is just the release of energy from glucose. Where did that glucose come from? Well, it came from photosynthesis. So we break it down and we release carbon dioxide. When you look at this equation, and I really do think it's a good idea for you to know the photosynthesis and the respiration equations. What do you notice about the relationship of this equation to photosynthesis?
It's backwards.
Yeah, they are opposite of one another. Okay, so what you see is the product here is the reactant side of photosynthesis and vice versa. Okay, did most of you already know that? Yeah, I thought so. Just wanted to make sure. Okay. There is a table here that comes out of your book that I liked, that just compares photosynthesis and respiration to each other. And so I'm not going to go through it, but I just think it's a nice little thing to kind of remind yourself of how these two things interplay with one another and kind of function to keep things living on our planet. Okay, let me stop the screen share so I can see y'all better. I have a feeling that there's not a good reason for us to move forward just yet into the algae today. Let's do that Wednesday because we don't have class Monday, right? We don't have lab either. By the time y'all get back, you're going to forget what lab looks like. Akira, you laughing at me? Do y'all have any questions about photosynthesis? In your studying, one thing I really would majorly suggest would be to draw out the processes for the 2 steps of photosynthesis. And you don't have to go into the detail that you see on your screen. You can go in just to the detail that we talked about, okay? Do you need to know the 3-PGA, the G3P? Not exactly. I need you to know that there's a 3-carbon molecule because that's where we get the C3 name for that version of photosynthesis. And we need to know that there's a number of steps between there and the 3-PGA, sorry, the GA3P. And why do we know the steps? Because that's where NADPH and ATP drop off. Y'all probably feel like you're being acronymed to death, and I trust me, I feel it too. I understand. But hopefully you get what I'm getting at, okay? I want you to understand the process, not necessarily the little minor details of the names, Do you understand how the Calvin-Benson cycle works? Okay, not necessarily can you tell me word for word which molecule is where. Okay, that's the detail that I want you to walk away in botany from. Okay, and of course, the whole purpose of the end of photosynthesis, and especially the end of the Calvin-Benson cycle, is to make that extra Ga3P, which becomes all of your glucose, right? Or other starches, however they get used. Those things then get turned into something that the plant uses. And also, if we eat it, or if a plant needs to break it down for themselves, then they're going to go through cellular respiration, right? So if you can just take the general photosynthesis, the C3 photosynthesis, and understand how the two steps work and how they relate, and then how does respiration relate, okay? And then I would kind of go into the more details, like how does photorespiration pop in, okay? Which I didn't give you a lot of details there, okay? I didn't tell you how photorespiration works. I just told you that it happens and that it ends up costing more to keep the plant alive, okay? If we want to avoid photorespiration, then we start looking at the variations on photosynthesis like C4 and CAM. Now I'm going to make a big point here that I forgot to make when we were going through the slides. The first step of photosynthesis is no different for all three, C3, C4, and CAM, all the same. It's just how do we get the carbon to the Calvin-Benson cycle that things change. So it's not even the second step of photosynthesis is different. It's just how do we absorb carbon and when do we absorb carbon and how do we get it over. That's the only difference really. So it's not what you learned at the beginning, step 1, step 2 of photosynthesis is the same. It's just kind of in between those two steps. How do we do it? Okay, that's the last thing I'm going to say. Any questions, comments?
When did you say that our test was going to be again?
I don't think I did.
Okay.
I want to get through algae.
Okay.
Okay, so it's not going to be next week for sure. Okay.
What about the lab quiz is probably going to be the next week then, that Monday.
So lab quiz will be when y'all come to lab for the next, rest of the semester. Okay, except for maybe like going out to Plymouth Bluff, possibly. You won't have one. Okay, and since we've missed a Monday, we're going to have to adjust some things a little bit, but we can talk more about the lab next week. The test 1 is going to be the following week sometime. So think about when y'all want to take it. But we have to get through algae before we can be able to take it. Okay. So we'll see how it goes next week. Okay, anything else? Okay, I'm gonna stop the recording